Yeah, so I thought I would give a pitch on Jørgen and I's, as Ditte just mentioned, our technology. I don't know if it's our technology, but at least we got this Cosmics instrument last year, a bit more than a year ago, actually, and that's where this picture was from. So back then, the instrument was installed in my lab in the Skov building at Aarhus University. And we got this instrument thanks to a grant through a consortium called CELEX that I guess most of you knows about. It's a national research platform dedicated to single cell and spatial omics analysis. And the Cosmics can do spatial analysis both at the RNA level and at the protein level. And I'll, of course, get back to that. So first, I just want to mention a little bit about how Jørgen and I, the smiles you see here, they were kind of wearing off fairly quickly because right after we were up and running, we had the training on the instrument and everything. There was this trial case between NanoString and 10x that actually prevented us from using the instrument for a long time because there was a ruling against NanoString that they could not service the instrument and they could not sell reagents, and we basically couldn't do anything with it until here in spring, NanoString appealed the decision, and then we were suddenly back in business, you could say, and could use the instrument again. Then what happened was that my technician in the meantime that had training in the instrument got a job in Copenhagen. So we had the training again, and at the same time we decided actually to move the instrument to MoMA, to the core facility at MoMA, where they already have a lot of NGS equipment and also the GeoMX from NanoString. So now our instrument is placed at MoMA together with Kasper Thorsen, and is in good hands out there. And we had the training again, so we are up and running again now. So just a little bit about the technology. So it is, as I said, a spatial technology, and it's based on hybridization. So the slide you see here is how it works at the RNA level. So basically what is designed are these relatively small probes. You can see in blue in this slide here, 35 to 50 nucleotides. And this probe has attached to it this so-called readout domain that is then undergoing a number of cyclic hybridization events where a lot of fluorescent reporter probes are hybridized, and then due to photocleavable linkers and UV light, they can be cleaved off again. And then you can imagine then the next, the dark blue readout domain. I'll just see if I can point to it. Here then a new hybridization event will take place, bringing in a new color. And then when this is done through a round of cycles, then you will build up a unique barcode for each unique RNA target that you have designed probes for. And similarly for the protein level, it's basically the same principle, only differences that instead of a specific probe, you have a specific antibody for the proteins that you are interested in. And then it works the same way. So the next couple of slides here are from NanoString's homepage, and it's just to give you an idea how an experiment is built up. So the first thing you see here is just an FFPE raw tissue, and then as part of the process, I didn't mention before, but there's also an immunofluorescent labeling of the membranes, and also there's a DAPI staining, and you can also have some immunofluorescent labeling of other targets to help visualize the tissues a bit better. So this is what you see here in the next slide, where you both have a membrane staining and in this case also a staining for epithelial cells. And then you use this information, or actually it's the instrument that uses this information to define all the boundaries of the single cells. So it's supposed to do a very good cell segmentation. And so far in our own experience, this is also the case. And I'll come back to that a little bit later. So when you have, for instance, information on 1,000 RNA transcripts and you have the cell boundaries, you know how many of each transcript you detect in each cell. You can also use that information to cell type and map all the different cell types in your tissues, as you can see in this slide here. So before I just talk to you about some of our own experiences and some of our own results, I just want to give you an idea about what is currently possible with this technology. So NanoString has a number of different panels that I'm listing here in this slide that you can purchase off the shelf. So they have recently launched a 6,000-plex panel for human, and then they have different 1,000-plex panels for mouse and human at the RNA level. And for the protein level, the plex is not as high, and they have a focus on neuroscience and immuno-oncology primarily. NanoString already presented the whole transcriptome for the RNA analysis at a conference earlier this year, and this will be commercially launched next year, according to NanoString. And it's also possible to do some custom add-ons. So at the RNA level, with the 1,000-plex panels, you can add up to 50 custom targets that you decided on yourself. And for the 6,000-plex, it's possible to add up to 200. But at the moment, I think this is not very feasible to have that many custom targets for most projects, because in my opinion, it's too expensive, honestly. And at the protein level, I think, I don't remember correctly, but it's around, I think, 8, something like that, custom targets you can add to these panels at the protein level. So I also wanted to show you a little bit of our own experience with this technology. So just for you to have an idea of what I'm going to show you, I'll give you a little bit of background for this project, which was on colon cancer. And in colon cancer, there's oftentimes a quite clear invasive front of the tumor where cancer cells can be sort of budding off from the main trunk of the tumor. And these small clusters of cancer cells or single cancer cells are called budding cells. And it is believed that these cells are kind of the first step in the metastatic cascade. So these cells are on the way to metastasize, is believed. So we wanted to use this technology to really focus on these budding cells. So we set up a project using CosMx on some colon cancer tissues. And here I'm showing you how we set it up. So we ran just a single slide on the CosMx instrument, but to lower the cost per sample, we had, as you can see here, we were supposed to have 11 samples, but one fell off. So sorry, we were supposed to have 12. So we were ending up with 11 samples here. And then the way the instrument works is that it it analyzes a defined area, so-called field of view, at a time. And you can place as many fields of views as you want, basically. And here you can see how we placed fields of views in these samples. And then we ran the 1,000-plex panel at the RNA level, and we also had these immunofluorescent stainings for membrane and for the nuclei, and then we also stained with pan-CK, which is epithelial marker, and then we stained CD45 and CD3 as well in these tissues. And this is just a very brief overview of the data we got out of it. So in general, we got more than 200 transcripts per cell for each field of view. And in total, we got around 30 million transcripts. So that's quite nice. Actually, for our project, we selected the fields of view with less than 200, but we could also have analyzed those, I believe. But just to have a cutoff and analyze only the fields of view that performed the best. So since we were new to the technology and the technology was very new at the time, we just wanted to start out with adjacent normal tissue to see how well the instrument actually performs in these very ordered and well-structured tissues. So what you see here are the crypts of adjacent normal colon tissue where pan-CK is staining all the epithelial cells. And then you have some yellow staining, as you can see, are T cells in the stroma in between the crypts. And then the blue is DAPI here. And then we ran, as I said, this 1000-plex Cosmix panel. And based on that data, like in single cell analysis, we can do these cluster analysis or UMAP analysis on all this data to unsupervised categorize the cells into different clusters. And what you can see here is that in this analysis, the algorithm made like two superclusters, one over here to the left and another one to the right. And when we look at the genes that are characterizing these cells, it was immediately apparent to us that the cells to the left here are all the epithelial cells, and then all the immune and stromal cells are the ones clustering over here. So we were actually quite happy when we then mapped this back into space, because for each of these single cells, we have the information where they are in space. And then they are just, again, unsupervised, mapped back into space. And you can already see these structures, the crypts of the epithelial cells. And we can overlay this information with the pan-CK immunofluorescent staining and see that it maps really precisely to these cells or areas of pan-CK staining as we expected from the gene expression patterns in these cells. And we can zoom in further and look at a single marker that are very specific for epithelial cells here, a gene called PIGR in this case. So also, another thing we looked at in the normal tissue was the T cells. So here I'm zooming in on an area where we had absolutely no staining for T cells in the immunofluorescent staining, and another area where the T cells were quite abundant, as you can see here. And actually, based on the 1000-plex gene expression panel, we didn't call a single T cell in this area where there was no staining, but we did call quite a lot of T cells in the other area down here. And we had a quite good overlay with what was called as T cells by the unsupervised algorithm and the CD3 staining, as you can see in this. I hope you can see in this small video here. So with that, we were very keen to move on to analyze the cancer tissues, of course. And this is actually one of the first kind of preliminary analysis where at the top you can see how the cells were called based on the unsupervised clustering algorithm, like I showed you before. And below you can see the pan-CK staining of exactly the same areas. And what you might appreciate from these pictures here is that all those blue and orange cells up here are staining positive for pan-CK down here. So these are the cancer cells. And then we have, of course, in the microenvironment out here, we have cells that are part of the tumor microenvironment, and we also have some cells, small clusters of cancer cells that are staining positive for pan-CK and also called as cancer cells up here. And in the middle of the cancer cells, for instance, here we have two cells that are as you can see here, pan-CK negative, and these were also not called as pan-CK, as cancer cells by the algorithm. So we think the instrument is doing a good job in calling the cell types and also defining the cell boundaries. And I didn't mention, but what I've turned on here in the analysis is the PICR gene that you see. All the signal for the PICR gene here is also quite specific for the cells that stain positive for pan-CK, all the cancer cells. So for instance, here you don't see a single detection of PICR in these two cells here. So analyzing this a bit further, we actually, the very first field of view we dived into, again, we saw these two superclusters of cells in the UMAP analysis, all the cancer cells to the right here and the tumor microenvironment cells to the left here. And when mapping this back into space, we saw that most of the cancer cells were located together, as we would expect if there's a clonal evolution going on in these tissues. However, one of these cancer cell clusters behaved completely differently. And at first we were a little bit puzzled about this. It looked a bit random where these cells were found, but upon closer inspection, we found it actually quite interesting that they were always in the outskirts of the tumor masses and oftentimes encompassing these budding cells, as I talked about before. So we wanted to see what is specific for all these different cell types. And in this heat map here, you can see the 10 genes that are defining each cluster mostly. And I highlighted here the cancer cell cluster that behaved very differently from the other cancer cell clusters in the spatial analysis. And what you can appreciate here is that they have higher expression of stromal markers and lower expression of some of the epithelial markers. Also a bit lower expression of the proliferation marker MKI67 and higher expression of this Wnt2b, which is known to induce epithelial to mesenchymal transition, or EMT, which has already been suggested in the literature that these budding cells are undergoing EMT, where they basically become more mesenchymal. So that's in line with the stromal markers going up and some of the epithelial markers going down. So we thought that was very interesting, and now we are also analyzing these data further, and we have of course analyzed many more fields of views, and we see the same intriguing patterns. And when we look closer at what are these the nearest neighbors of these cells, very consistently we found that these cells are more often than would be expected by chance in contact with stromal cells that we believe are cancer-associated fibroblasts. And in this analysis, we can also look at receptor-ligand interactions and see at the RNA level how cells are potentially talking to each other. And we found one very interesting receptor-ligand interaction that I'm highlighting here that was consistent across all the fields of views in the cancer cells being upregulated. And I don't know if you can see it, but each of these dots here for each of the fields of view represent a cancer cell cluster. And the cancer cell clusters with the budding cells are the blue one. So the blue one is almost every time on top in this plot. And then you see this interaction doesn't occur very much in the healthy tissue. So we found that quite interesting, and we believe that this receptor-ligand interaction could facilitate these cancer cells being integrated into the tumor stroma and be facilitating in the end the path towards metastasis. So by the end here, I just want to mention a little bit more about how we can analyze the data. So all the plots I showed you so far, Juan Rodriguez from my group has prepared using genome DK, so a little bit more, you could say, hardcore bioinformatics, and using a lot of scripts that are also well known from the single cell analysis. You can also analyze and visualize data directly in this Atomics platform. So this is from one of our recent experiments where we did the protein panel. And what you can see here in this visualizer, we can zoom in and out of the tissue, and we can toggle on the different proteins that we have in the panel, also the immunofluorescent stainings. We can decide to have the cell segmentation on or off. You can see here now it's off in this picture here, where you have pan-CK in green again, like before, staining all the cancer cells. And then what I turned on here in purple is the proliferation marker Ki67. And this I turned on just that you can see some cells are actively proliferating here and down here. And what you can also see is that this is very specific for the nuclei. So in this analysis, you can really do subcellular analysis as well and clearly see if something is in the nucleus or in the cytoplasm. And then again here in yellow, I turned on a marker for T cells, and in red, a marker for fibroblasts in the tumor microenvironment. But there's a lot of things, of course, you can do with these types of data. Of course, we are just getting started again now, as I mentioned in the beginning of the talk. And Jørgen and I, we are very open to collaborations using this technology. So I just wanted to put a slide here with our contact information. You are very welcome to contact Jørgen or me if you have an ODIN project where you think this technology might come in handy. And then lastly, before I take any questions, I just want to show my acknowledgement slide, and in particularly highlight Juan here, who I mentioned before, who did almost all of the analysis I showed you in the presentation here today. And I'll be happy to take any questions.